Alrighty. Welcome everyone. Hope you are having a good night. We have some cool things in store tonight. So my plan let's see. My plan tonight, I'm going to go over the phylogenetics and the science around SARS-CoV-2. That should be really cool and interesting. I have like 16 papers that I need to go through. So it is going to be packed. We're going through everything. My goal is to basically go through the papers in regards to phylogenetics, origins, all that kind of stuff. So it should be a pretty fun episode. Pretty fun ideas. I'm just trying to get everything set up, so it's my bad. Sorry, I am not good at this. All right. But to start off the night, I have something really cool to share. I don't know if I want to hold it now or till at the end, which is kind of exciting. Okay. Okay. Let's go through it. Oh man, I was muted the entire time. That's exciting. Professional streamer. I'm curious like how it's going to do video by video or everything kind of all together. I don't know. We'll just kind of see. We'll just kind of go through it and we'll see. Okay, so the first paper that I have. All right, the first paper. Emergence of SARS-CoV-2 through recombination and strong purifying selection. All right. Okay. Dang, some of these papers are so cool. I can't wait to read them. I read them like I've skimmed them before, but I want to read them more. Warby. This is like a perspective, so it's a little bit different than his actual full paper. Here's Picard. That's going to be a good one. This is the full Warby. So this looks like he. Oh my gosh, 300,000. Picard's at 153. Okay. Okay. Cool. All right. So the first paper we have Li et al. 2020. So this is Scientific Advances in July 2020. So we're trying to look at the actual science data on the phylogenetics, everything that has to deal with SARS-CoV-2, where it came from, all that kind of stuff. So I'm interested in any type of like sequencing data or any type of thing that we can learn about where it came from. So Li et al. 2020, July 2020. So pretty early on in the pandemic. If you remember back. The pandemic event with the Wuhan wet market and all that kind of stuff is November, December 2019. So this is a little bit after this. So here we go. COVID-19 has become a global pandemic caused by the novel coronavirus SARS-CoV-2. Understanding the origins understanding the origins of SARS-CoV-2 is critical for determining future zoonosis, discovering new drugs, and developing a vaccine. So, as we've talked about previously, where there has been a lot of need to future zoonosis, where did SARS-CoV-1 come from? Where is SARS-CoV-2 coming through? Discovering new drugs. The idea is like, well, if there's things that we can figure out the mechanisms of how this coronavirus is working, maybe there's specific drugs that we could use, and then develop the vaccine. In that, once we have a consensus sequence, then we can work on creating like a vaccine for that. We show evidence of a strong purifying selection around the receptor binding motifs, RBMs, in the spike and other genes among bat, pangolin, and human coronaviruses, selecting similar evolutionary constraints in different host species. We also demonstrate that SARS-CoV-2's entire RBM was introduced through recombination with coronaviruses from pangolins, possibly a critical step in the evolution of SARS-CoV-2's ability to infect humans. Purifying selection in different host species together with different recombination among coronaviruses suggests a common evolutionary mechanism that could lead to new human coronaviruses. So the idea being that, recombination isn't just a single mutation event. These are separate coronaviruses that are infecting the same host, and then they are actually changing their genomes and intermixing, and then that's causing a brand new, a new strain. Okay, so let's look at the results. SARS-CoV-2 recombination with pangolin, so this is pangolin, and bat SL. I don't know what SL means. SCOV. Simplot genetic similarity between SARS-CoV, Wuhan 1, and representative CoV sequences using 400 base pair window at a 50 base pair step and the Kimura 2 parameter model. So this is for... Okay, so this is looking at the genome similarity. Here's SARS-CoV. So this is blue. Wow. They're super similar. I don't even know which one specifically they're... I think it's the dark blue. Interesting. I think, yeah. So there's different binding mutations here at these spots. That must be like where the cleavage sites are. Or so this is the ORF1, ORF1A, 1B. I don't know what those are specifically. Oh, here's SARS-CoV. Boom. So it's this one right here. Really distant similarities there. But they are very similar. They track very similarly. Here's that ACE2 binding domain. So again, very similar to these. So these are phylogenetic trees that are made to basically the ACE2 binding domain, showing here that they have a huge clump in aggregate between the pangolin, SARS-CoV-2, and the pangolin, and these ones too. The SARS-CoV. I wonder if this is SARS-CoV-1. Okay, so this is an alignment now. So this is basically all the different amino acids that are in that binding motif, and there's a lot of them. But these are parts that aren't the same. So this is SARS-CoV-2 at the top, and then these are the chunks that are missing. So these would have to be the parts where the new mutations, the new, it's not mutations, the recombination events are actually filling that in. Dashes indicate amino acids, dots indicate deletions. Yeah. So there's some type of event where it filled those in. There's a structural analysis of this, of the more ACE2 binding spots. I wonder. I want to just get to the link real quick. This is Lee. Where's Lee? Okay. Strong purifying selections Lots of SNPs, dude. Lots of SNPs. But I mean, the big ones are these huge differences between the bat coronaviruses and the SARS-CoV-2 at the top here. So it's a huge difference. And this is like that would be the Furin cleavage site, must be, because this is a receptor binding motif. I don't understand what they're saying. Strong purifying selection. Strong purifying selection after Furin cleavage site and S gene among the SARS-CoV-2 and closely related viruses. Phylogenetic tree left, so up here, and highlighter plot right of the sequences around the RBM, the receptor binding motif, and the Furin cleavage site compared to SARS-CoV-2 Wuhan H1. ACE2 receptor binding motifs and the Furin cleavage site highlighted in gray boxes. So there's the non-synonymous. Mutations compared with the one are light blue for synonymous and red for non-synonymous. Dominance of synonymous mutations within group 1 compared with group B highlighted on the right. Position numbers are counted in number of nucleotides NA from the beginning of the region. Negative selection with groups. So this is showing here's the Furin cleavage site. You have a bunch of non-synonymous mutations, I guess. So you have, why would they be negative selection within groups? Interesting. If it was a purifying selection, I would thought that they, like, aren't they talking about like a kind of like a sequence that would want to increase in the population? So what they're saying is that it gets so weird and gets so different that it just, those differences in the mutations kind of stay. Because these are the SARS-CoV-2 at the top. And as you go genetically separate from it, you create more red. That would be the hypothesis, right? And more non-synonymous as you go away. Or indels. Cumulative plots of each codon average behavior for all pairwise comparisons for indels, synonymous light blue and non-synonymous red by group. The abrupt slope change of the non-synonymous curve in group A at codon 368 is indicative of a shift in localized accumulations of non-synonymous mutations after the Furin cleavage site. Group B instead lacks this abrupt change in slope at the same position. Interesting. The abrupt slope change of the non-synonymous group A 360. So that's what this line right here is indicating, this big jump up. Is indicative of a shift in localized accumulations of non-synonymous. Oh, so it's that red that jumps up there, and that doesn't do that in group B. Group B instead lacks this abrupt change in slope at the same position. Values of W, I don't know what that W is, denote average ratios of the rate of non-synonymous mutations after the Furin cleavage site. The group B instead lacks this abrupt change. No. And then it's the derivative of that. position numbers are counted in number of amino acids from the beginning. Sequences DS-DN ratios compared to Wuhan with codons 1 through 368 and codons 369 to 620. So it's this abrupt change, this recombination event that doesn't align the two genomes and the two groups. Then I think that's what they're trying to say. Now they're looking at each of the parts of the genome. Dang, this is interesting. Purifying solution. There are three important aspects of beta coronavirus evolution that should be carefully considered in the phylogenetic reconstructions among more distant coronaviruses. First, there's extensive recombination among all of these viruses, making standard phylogenetic reconstructions based on the full genomes problematic, as different regions of the genome have distinct ancestral relationships. Second, between more distant sequences, synonymous substitutions are often fully saturated, which can confound analyses of selective pressure and add noise to phylogenetic analysis. Last, there are different selective pressures at work in different lineages, which is worth considering when interpreting trees. The current sampled coronaviruses are too divergent from SARS-CoV-2 to be its recent progenitors, but it is noteworthy that these sequences contain a receptor binding motif that can most likely bind to human ACE2. That's huge for like recombinations, right? So it could like probably recombine. While rat G13 is the most closely related CoV sequence to SARS-CoV-2, it is distinctive. It has a distinctive RBM. In addition, a recent study showed that rat G13 pseudovirus is much less effective than SARS-CoV-2 pseudovirus in using ACE2 to infect cells. So they put the rat G RBM into a SARS-CoV-2 coronavirus that we could genetically alter, which is a pseudovirus. It's probably an adenovirus vector or something like that, putting the that sequence, that receptor binding motif, into another virus to try to see if it could actually infect the cells. SARS-CoV-2 was a nearly identical RBM to the one in the pangolin CoVs from Guangdong. Thus, it is plausible that rat G13 like bat CoV viruses may have contained the RBM sequence binding to human ACE2 through recombination with pan-SL-CoV-2 GD-like viruses. Okay, so yeah, it's that recombination event that's important. We hypothesize that this and other ancestral recombination events between viruses infecting bats and pangolins may have played a key role in the evolution of the strain that led to the introduction of SARS-CoV-2 into humans. There you go. It is possible that other not yet identified hosts infected with CoVs that can jump to human populations through cross-species transmission if they can successfully infect humans through the ACE2 or other receptors. An analysis of 6,400 SARS-CoV sequences from GISAID identifies only one very rare mutation, G4576S, that is direct ACE2 contact residue. It was found in a local cluster of sequences from the Washington state. You'd think that there would be more. But you'd think that there'd be more, right? If it was needed or really good for that transmission. However, it is at the periphery of the receptor contact surface and so not significantly affect the binding affinity. Okay. All three human CoVs, SARS, MERS, SARS-2, are the result of recombination among CoVs. Recombination in all three viruses involved The S gene, likely a precondition to zoonosis that enabled efficient binding to human receptors. Oh, what does the S gene make again? What is that? S gene SARS-CoV-2 function. What is this? Oh, dude. Of course, it's the surface glycoprotein. Yeah. Because it's an enveloped positive sense thing. So that's the. Oh, okay. The structural proteins of SARS-CoV-2 include the envelope protein, the spike or surface glycoprotein. Ah, there it is. That's the spike. The spike. Derp, derp, derp. Okay. 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 We're getting back to it. Now I can remember this stuff. Okay. The membrane protein and the nucleocapsid protein. Spike protein is found on the outside of the virus particle and gives the coronaviruses their crown-like appearance. Gotcha. And this is the biochemistry of it, how it actually is made. This is the life cycle of it. Interesting. Oh, cool. That's really cool. That looks awesome. Okay, cool. Always down for some proteomics pictures. I can't I can't look at them at all. I have no idea what we're looking at, but they look cool. Why S-MAP gene sequencing is key for SARS-CoV-2 surveillance. It's the surface protein, the spike protein, which is the homotrimeric glycoprotein complex essential for infectivity. Okay. Gotcha. That makes sense. All right. So there has to be some recombination, some major mutation event in order to kind of like see this thing happen or go through. Okay. Making more sense as we go along here, isn't it? Okay, back here. Extensive recombination among bat CoVs and strong purifying selection pressure. I just want to purifying selection. I just want to make sure we're understanding this. Purifying selection. Oh, it reduces genetic diversity. Yeah, that's what I thought. That there was going to be that sequence is going to show more prevalence in the population. Natural selection, purifying selection is the selective removal of alleles that are deleterious. This can result in a stabilizing selection through the purging of deleterious genetic polymorphisms that arise through random mutations. I mean, that would make sense, right? Because they're actually getting in and infecting. So as they kind of make that jump, only the ones that have that beneficial mutation are going to go. So any type of random mutations are going to kind of be pushed aside, right? Okay, cool. Learning new terms and relearning some. Okay, strong purifying selection pressure among viruses from humans, bats, and penguins may allow such closely related viruses to readily jump between species and adapt to new hosts. Many bat-CoV-Vs have been found to be able to bind to human ACE2 and replicate in human cells. That's scary. Serological evidence, which is the antibodies found in blood, serological evidence have revealed that additional, otherwise undetected spillovers have occurred in people in China living in proximity to wild bat populations. Whoa! There's been spillover events that have happened before with other coronaviruses. Current surveillance of coronaviruses in the hosts in humans will likely. All the SARS and MERS originating strains have been found in civets and dromedary camels, respectively. Wow. So far, efforts to identify the close link in the original pathway of SARS-CoV into humans have failed. The new SARS-CoV-2 strain did not cause widespread infections in its natural or intermediate hosts, then such a strain may never have been, then it never be identified. Oh, that's a really interesting point. Because if the if the recombination event happens, how long did that new how long did it happen in nature before it actually spilled over into humans? So what they're saying is that there needs to be widespread infections in bats and pangolins, and for that intermediate host to actually create a bunch of new ones, new strains, so that we can track it genetically. If it was like a spillover event and then that crossed and then went right into humans, that's not a good thing for tracking it, you know. Close proximity of animals of different species in a wet market setting may increase the potential for cross spillover infections by enabling recombination between more distant coronaviruses and the emergence of recombinants with novel phenotypes. While the direct reservoir of SARS-CoV-2 is still being sought, one thing is clear: reducing or eliminating direct human contact with wild animals is critical to preventing new coronavirus zoonosis in the future. Yeah. Good luck, man. Good luck with that one. That's going to be rough. Okay. So now it kind of like makes sense now more. So it makes the synonymous, if in a deleterious stuff is going away due to purifying selection, then our hypothesis would be that the synonymous mutations would increase over time because all the deleterious mutations are going out, and you're basically purifying or creating more and more of the same sequences. And that's what they find. This cumulative behavior over not over time, over the sequence of the receptor binding motif. Very cool. Okay. So very strong. What did we learn there? That's very strong evidence to show that there's bat pangolin recombination events that have happened before. We know that it's happened in other times in humans. It's just that it wasn't widespread. Or it was widespread because that's how we know about it, but this hasn't happened. Interesting. Okay. Host-specific asymmetrical accumulation of mutation types reveals the origins of SARS-CoV-2 is consistent with the natural process. That seems very pertinent. The innovation. I don't have these. Let's see if we have any 2020 papers. Okay, let's go down the line for 2021. The asymmetric de novo mutations Okay. Next, Shan et al. 2021. So August 30th. Post-specific asymmetrical accumulation of mutation types reveal that the origin of SARS-CoV-2 is consistent with the natural process. The asymmetrical de novo mutations, so new mutations arising in SARS-CoV-2, are induced by mutagenic mechanism in the host cellular environment. De novo mutations determine the molecular spectrum of accumulated mutations during SARS-CoV-2 evolution. So where did the mutations come from that exist within SARS-CoV-2? Molecular spectra of accumulated mutations in beta coronaviruses cluster according to the host species instead of the phylogenetic relationship. Molecular spectra of accumulated mutations in beta coronaviruses cluster according to the host species instead of the phylogenetic. So the mutations that are selected for in the beta coronaviruses are similar because of the host species and not because of how closely related they are phylogenetically. What is the difference? Are all beta coronaviruses? What's the difference between a beta coronavirus and a regular and the other coronaviruses? One of 4 genera of coronaviruses. It's one of 4: alpha, beta, gamma, or delta of coronaviruses. Member viruses are envelope positive strain viruses that infect animals. The natural reservoir for beta coronaviruses are bats and rodents. Okay. So there's different families of them. What are these? What's the difference between these two? Oh, okay. Gamma cove are known as the coronavirus 3. Okay, so it's just like where they infect. While the alpha and beta genera are derived from the bat gene pool, the gamma and delta genera are derived from the avian and pig gene pools. Okay, so this is what it's talking about. That they are hosts instead of the... Okay, interesting. The mutations accumulated in SARS-CoV-2 prior to its transmission to humans are consistent with the evolutionary process in a bat host. Okay, so this confirms what we were talking about before, right? The mutations accumulated in SARS-CoV-2 prior to transmission to humans are consistent with the evolutionary process in a bat. The capacity of RNA viruses to adapt to new hosts and rapidly escape the host immune system is largely attributable to de novo genetic diversity that emerges through mutations in RNA. So the capacity of these RNA viruses, where do the mutations come from? And how do they adapt to new hosts? And how do they accumulate those mutations in order to evolve to escape the host immune system? Well, they're new mutations, and that mutation happens through RNA synthesis, probably, because they have to make new RNA when they're in the host. Although the molecular spectrum of de novo mutations, which is the relative rates at which various base substitutions occur, are widely recognized as informative toward understanding the evolution of a viral genome, little attention has been paid to the possibility of using molecular spectra to infer the host origins of a virus. Here, we characterize the molecular spectra of de novo mutations for SARS-CoV-2 from transcriptomic data obtained from virus-infected cell lines enabled by the use of sporadic junctions formed during discontinuous transcription as molecular barcodes. Characterize the molecular spectra of de novo mutations. So they're looking at how the new mutations are created by looking at transcriptome data, which you kind of have to do, right? You can't look at RNA. It's a positive sense. You have to go by that. Here we characterize the molecular spectrum of the de novo mutations for SARS-CoV-2 from the transcriptome data obtained from virus-infected cell lines, enabled by the use of sporadic junctions. I don't know what this means. This is really cool, though. Enabled by the use of sporadic junctions formed during discontinuous transcription as molecular barcodes. I'm excited to see what this is. I don't know what it is. We find that de novo mutations are generated in a replication-independent manner, typically on the genomic strand, and highly dependent on mutagenetic mechanisms specific to the host cellular environment. So the new mutations are generated in a replication-independent manner, typically on the genomic strand, and highly dependent on mutagenetic mechanisms specific to the host cellular environment. I'm confused what that means. Find that the new mutations are generated in a replication independent manner. So it's not about when they replicate their genome? And highly dependent on mutagenic mechanisms. So that must be just like ways that the cellular environment is creating mutations. Normally that's like UV or stuff like that. How would like a chemical, like chemically, nucleic acid nucleotides are changing? De novo mutations will then strongly influence the types of base mutations accumulated during SARS-CoV-2 evolution in an asymmetric manner, favoring specific mutation types. Consequently, similarities between the mutation spectra of SARS-CoV-2 and the bat coronavirus rat G13, that keeps showing up, rat G13, which have accumulated since the divergence, strongly suggest that SARS-CoV-2 host evolved in a host cellular environment highly similar to that of bats before its zoonotic transfer into humans. Oh, so they looked at the mutations that accrued in this de novo sense, and they compared it against the environmental phylogenetic, like the genetic sequencing that they have, and they look very similar, those mutations. So consequently, similarities between the mutation spectra of SARS-CoV-2 and the bat coronavirus ARC-G3, which have accumulated since the divergence, strongly suggest that SARS-CoV-2 evolved in a host cellular environment similar to that. So, oh, that's really interesting. So rat G13, the differences between them, because this is the most highly related one. So what are the mutations between them, and where do those mutations come from? The random mutations accrued from this cellular environment mutagenesis, and not just an RNA polymerase. That's really interesting, because if it was in a lab, then you wouldn't be getting those cellular environments necessarily, because you'd be doing a lot of like in vitro transcription and stuff like that. So that would be polymerase errors, and those would be different types of errors. And they're saying that they didn't see those. Wow. Dude, that's cool. That's way cool. No way. How did they do this? Collecting graphical abstract. Dude, people are so smart. Like, I'm not kidding. Sometimes I'm like, this is unbelievable. This is so cool. Okay, so the SARS-CoV-2 transmission, divergence from bat coronavirus rat G13, right? So, that happened in 1969? And the outbreak in 2019. Where did those mutations come from? Molecular spectrum of mutations. I'm looking at rat G13 and then looking at it where it is now, you can kind of see that, okay, it had to be in a similar. Okay, I want to go to figures first. Shout out to Shan et al. What a cool idea. Let's see what this is. Okay. RNA, the barcoded by via circulation, rolling circle reverse transcription. Okay, so they're making more by this type of rolling circle transcription rates, and this is going to be errors that accrue. And then we look at it again in error correction, looking at where did the errors happen. RepSeq, which would be the RNA barcoded by RNA oligos. Oh my gosh. They bound them together. I thought that they would use reverse transcriptase to make it DNA and then do it. But they actually use RNA oligos, and the reverse transcriptase just would reverse transcribe them all together. How are these barcoded oligos binding to these RNAs? They read with an identical barcode. It must be this mutation. They must be like binding to the specific mutation. Junction barcoded subgenome transcription multiple times by RDRP. So this is restriction RNA dependent RNA polymerase. So this is RNA. It's made from RNA, which is kind of cool. And this would reads with identical junction or read family. I usually like to look at the figures, see what I want to see what's going on before I go back and actually do it myself. Junction barcoding, conventional approach, junction barcoding approach, mismatch frequency, lower number of detected de novo mutations in SARS-CoV-2. Molecular spectrum of de novo mutations in SARS-CoV-2. Mutation rate, number of mutations. So they should be similar in similar hosts. So then they compared it. Among patients' polymorphisms, de novo genetic sequences, positive selection or genetic drift, negative selection or genetic drift. Dude, this is some gnarly shit. This is so cool. This is so cool. It's some like really applied molecular biology stuff here. This is amazing. Virus with virions. This is why it'd be so hard to fake this stuff. It's like these guys are coming at you with like the gnarliest methods that you've never even considered. Like, how could you? It would only make sense if it was one way. Oh, so they applied it to the mutations. Here we go. So isolated from humans, the different SARS-CoV, and then they compared them. And they line up perfectly. Human related, bat exclusive. Wow. Shan et al. That is so cool. Dang. That is some gnarly applied molecular biology. That is so cool. I'm so jealous of stuff like that. Those guys are so... and people, I should say, I don't know. What incredible applications, and I'm blown away. What an awesome paper. I've never read this paper before. I've never even thought or used that before. Wow. Let's read the discussion. Usually abstract discussions a good read. Figures are good. Let's see what they say. Okay. This should be good. I'm excited for this. I'm blown away. That is so cool. There's no way that you'd be able to fake that data. It would be impossible. There'd be no way you'd have, in order for stuff to be biotechnology made for SARS-CoV-2, you would have had to start this 50 years ago. It's like, according to this paper, basically, they're saying like the amount and where the mutations are and how they've accrued and what's happened is like how it happened in nature starting 50 years ago. So you would have had to been evolved somehow 50 years in the making. That's incredible. Damn. Damn. That is so fun. That is so fun. I love it. Okay. I'm excited for this discussion. The central dogma of molecular biology asserts that genetic information of cellular organisms is stored in DNA, which can be transcribed into mRNA for transmission of genetic information into functional proteins. No citation needed. Like, what would you cite for that? Well, that's true. This is true. We're talking about things that exist. And okay, although the presence of mRNA mutations has been confirmed in a few cellular organisms, they affect only a limited number of proteins due to the transient nature of mRNA. However, for RNA viruses whose genetic information is stored in RNA, mutations in RNA can have a long-term influence because such mutations have a chance of being inherited. In this study, we exploit this influence to infer the evolutionary history for RNA viruses, in particular SARS-CoV-2. That is such a cool idea, dude. That is nuts. Because genetic information gets passed down, not mRNA. So any mutations that accrued in an mRNA aren't going to accumulate in a population. Dang. That is so cool. Because the RNA viruses whose genetic information is stored in RNA, mutations in RNA can have a long-term influence because such mutations have a chance of being inherited versus mRNA where it's not. It is worth noting that 4 out of 5 branches that represented a mixed host history in bats and a non-human organism, pangolins in B5, B9, civets in 11, camels in B20, hedgehogs in B25, 25, fell into the 95 confidence ellipse estimated from the bat fell into the 95% confidence ellipse estimated from the 17 bat exclusive branches. So they're saying, like, why would these, if it's host specific in the cells, right, in the cellular environments and mutations accrue there, then why would these things accrue in these beta coronaviruses in these organisms? Plausible explanation is that the majority of the base substitutions present in these 4 bat mixed branches were accumulated in bat cellular environments in light of the unique cellular features reported in bats compared with other animals, i.e., high concentrations of endogenous antioxidants. So they have high antioxidants in them that are make for unique cellular environment. In other words, our approach can identify a host jumping event from molecular spectra only if sufficient mutations have accumulated in the new host. In other words, our approach can identify a host jumping event from molecular spectra only if sufficient mutations have accumulated in the new host. If something is identical and all the mutations are the exact same, then this approach isn't going to be able to find them, is what they're saying. In the future, characterization of molecular spectra of RNA mutations in additional species, especially in bats, using SIRSEC and RepSec, will be promising for tracing the transmission route of SARS-CoV-2. Some additional caveats to the conclusions drawn from our results are discussed there in the supplemental. Although this study focuses on the relative rates among base substitution types, i.e. molecular spectra, we estimate an average rate of 1.73 times 10 to the negative 5th de novo or new mutations per nucleotide in SARS-CoV-2. Despite the proofreading mechanisms provided by its non-structural protein 14, SARS-CoV-2 mutations still occur at a rate 3 to 4 orders of magnitude higher than DNA mutations, which enables rapid immunological escape while leaving genomic integrity maintained by natural selection for infectivity. In addition, our analyses focused on the detection of point mutations. Although not highlighted in the results, we also estimated the molecular spectra of indels, which is indels are insertions or deletions. Although not highlighted in the results, we also estimated the molecular spectrum of indels with the similar computational strategy that used junctions as molecular barcodes for the intermediate negative sense subgenomic RNA. Okay, this work identified 2 small insertions and 96 deletions, the majority of which are shorter than 6 nucleotides. Interesting. 96 deletions, shorter than 6 nucleotides. Interesting. Although the significant illness and death caused by SARS-CoV-2 and coronaviruses has led to multiple studies on this virus, basic understanding will not key features such as its origin. We show that the mutations accumulated in SARS-CoV-2 prior to its transmission to humans are fully consistent with a natural evolutionary process in a rhinoplus bat host. In addition to this theoretical purport, our methods will be useful to identify the natural hosts of other RNA viruses that can potentially be applied towards the prevention of future outbreaks. Shan et al. 2021. Amazing paper, dude. Wow. Dude, what are the metrics on this thing? Are you serious? No way. Oh, man. This is a great example of like, I don't understand why some papers just don't blow up, dude. I really don't. I mean, good Twitter engagement. But like the citations are really low. I'm really shocked, to be honest with you. 16 citations? I guess like the RNA sequencing approach and the molecular biology is very specific. So it'd be very like, it'd be hard to, who is this cited by? How is it not cited by more people? I mean, I guess it's just so novel. It's such a novel molecular biology approach that it's like a lot of people haven't used that yet. It's a new RNA based genotyping kind of thing. How? So it just kind of like behooves the people to use something that exists. Yeah, so it's basically where the question, the main thing is where did these 500 mutations come from? And comparing those two. As per the traditional, the absence of evidence is not the evidence of absence, especially when considering the vast number of unexplored wild animals, the even greater number of viruses they harbor. While the considerable efforts from many research groups to search in nature for a closely related coronavirus may yet provide many insights into the origins of SARS-CoV-2, we instead turned our attention to those 500 base pair substitutions that have accumulated in SARS-CoV-2 because we hypothesized they could provide an unprecedented statistical power. Wow. That is so, I mean, I'm blown away. That's a cool paper. That is a really cool paper. Okay. All right. So dissecting the early COVID-19 cases in Wuhan 19. So I think that this is like a paper. We're going to read the Warabee's like full paper coming up. So I think this is kind of like the impetus going into it. Virus Hunter, discovering the evolutionary origins of SARS-CoV-2. Okay. So they're talking about RNA samples, transcriptomics, alignment. Oh, that's quick. Why? Oh, it's a preview. Between the bat-born and pangolin-born coves. Interesting. I don't understand why you'd put something like this out. Dude, Rhinophilus vats. That's them. That was it. Horseshoe bats. That's it. We know it. Next. It's not enough, apparently. It's not enough. Zhou at all. How was that not the first one that he linked? This must be it. Identify. Okay, so it's this. It's this one right here. Do we have this? Identifying. I mean, this has to be in this. Yep, there it is. Okay, we're going. We're going. We're cruising. We're cruising. Okay, cool. Okay, so I guess the story so far, 2021. You know, we've discovered horseshoe bats. Have a good environment, a good cellular environment for these coronaviruses. There's host specific mutations that accrue because of the cellular environment of these things, and they similarly track of 50 years of evolution and accumulated mutations between rat G13 and the one that we have right now that we've identified. All right, and there's like pangolin and bat coronavirus recombination genetic elements that make sense, and they align for what we actually see. Okay. I don't want to do like single authors. If we can do full papers, that'd be nice. So the Furin Cleavage site is a part of much debate because the it's it's a really important part in that spike protein, and it also it has such a big deal with the transmittance of it. So it's needed in order to transmit on the spike. Okay. There's the Furin Cleavage site. So this is the part that is different and unique. I thought there could be oh my gosh. This is just eviscerating. This is nuts. Ugh. This is really okay. let's just cover it and talk about it. This is really, they're really frank in the language in here, so that should be fun. Okay. So compared with other SARS-related coronaviruses, SARS-CoV and SARS-CoV-2 possess a unique Furin cleavage site in its spike. This has stimulated discussion pertaining to the origins of SARS-CoV-2 because the FCS, or Furin cleavage site, has been observed to be under strong selective pressure in humans and confers the enhanced ability to infect some cell types and induce cell-cell fusion. Furthermore, scientists have demonstrated interest in studying novel cleavage sites by introducing them to SARS-CoVs. We review what is known about the SARS-CoV-2 Furin cleavage site in the context of its pathogenesis, origin, and how future wildlife coronavirus sampling may alter the interpretation of existing data. I don't know if I want to read the whole thing again. Okay, so it's required. The FCS has been deleted, and it shows that it's not effective anymore. Mutations impacting the FCS rate are high in humans. Less than there's specific parts that are really low deleted. How does SARS-CoV-2 spike compare with those of the other SARS? So this is interesting because there's so much talk about where did it come from. So I'm curious. Like I had heard that there is like just a simple recombination or something that would lead to it. So I'm curious if they talk about it here. Several novel SARS-CoV-2 like genomes from the horseshoe bats in Japan, Cambodia, Thailand, Laos, and China have been published. However, to this day, no known sarbevovirus except SARS-CoV-2 has an FCS insertion at the S1, S2 junction. To understand how unique the S1, S2 FCS in SARS-CoV-2 is, we realigned the S1, S2 regions of known SARS-CoV-2 like CoVs. We found that the alignment is sensitive to lineage sampling by visually comparing a codon sequence alignment of all known spikes to alignments of a few subset of the spikes. The full alignment appears to feature numerous insertions at the different positions in the S1, S2 region across the range of spike sequences examined. Thus far, the PRRA motif is unique in its position, whereas the spike sequence more distantly related to that of the SARS-CoV-2 present other commonly featured insertions at their positions. For instance, despite being collected from different locations, bunch of different types, appears to be conserved across those four more closely related spikes. It is currently difficult to know exactly how unique the SARS-CoV-2 PRRA motif is among SARS-CoV-2 like spike sequences because these viral lineages are relatively undersampled. Ah, interesting. Based on an analysis, an additional SARS-CoV-2 like spike sequence has the potential to reform and shift the alignment over this region and thus dramatically impact interpretations about the evolutionary history. To demonstrate this, we examined 3 alignments of the S1, S2 region created by excluding sequences that introduced indels in the full alignment. Removing SARS-CoV-2 alone was sufficient enough to change the predicted insertion positions among the remaining SARS-CoVs. Interestingly, further excluding other related ones resulted in another dramatic change in the alignment. In other words, our analysis showed that a single new SARS-CoV-2 like spike sequence from a lineage that has not yet been sampled has the potential to reshape the S1, S2 region alignment. That's interesting. That it would have so much of a difference. This is frustrating. Without broader geographical sampling of diverse bat species, we are left with an incomplete, probably biased picture of the evolutionary history of the spikes of SARS-CoV-2 like viruses. Some papers have proposed alignments that suggest insertions at the S1, S2 region in these strains. However, they did not result in the polybasic motif or an FCS. From our analysis, the alignments represent only one of numerous possible alignments. In our alignment, these quote-unquote insertions are located in different parts of the S1, S2 region. So they're just having these insertions aren't making this Furin cleavage site if they're proposed. So regardless, the alignment over the S1, S2 region is prone to revision when new sequences are discovered in the future, especially when so this is about sequencing. And sequencing that population, the difficulty of that. Nevertheless, due to how undersampled these viruses are across different host species, it is not straightforward to predict how much recombination and template switching might have occurred to result in SARS-CoV-2. There is an instance where the S1, S2 FCS was lost in a singular CoV, while its close relatives with similar spikes retain the FCS. However, this is the reverse scenario of that of SARS-CoV-2, which appears to have gained a highly advantageous FCS compared with the rest of its clade. Okay, we should read this. Okay, so Jack Nunberg, whose group first inserted an S1, S2 FCS into the spike of SARS-CoV, said, quote, "There is no way to know whether humans or nature inserted the site in SARS-CoV-2." In 2006, Nunberg's group had investigated whether the fusion activity of SARS-CoV would benefit from the proteologic cleavage similar to other CoVs that have been characterized at the time. They inserted a synthetic Furin recognition sequence at the putative R667 S1 S2 cleavage site and demonstrated that the FCS insertion, as compared with the substitution upstream of the R667, markedly increased the ability of the spike to induce cell-cell fusion. A group led by Gary Whitaker later showed that a substitution was sufficient when combined with the introduction of another FCS at the S2 region. This experiment in SARS-CoV was inspired by the observation of a similar setup in IBV strain, which had sufficiently cleavage of both S1, S2, and S2 prime. Whitaker recently, August 2021, published a comment describing the SARS-CoV-2 FCS as, quote, highly unusual. Similar studies on MERS-CoV had also determined that its FCS is required for a sufficient entry into human lung and intestine cells and influences the cell tropism of the virus. Yeah, I mean, you could just take that sequence and put it into another virus and then see basically if you can actually infect ACE2 cells. In September 2021, it was reported that an international group of scientists, including the Wuhan Institute of Virology, had in March 2018 proposed a roadmap for detecting novel proteolytic cleavage sites, including FCSs, in the spike sequences of novel Cerberoviruses and inserting these novel cleavage sites into the appropriate parental strain. These research endeavors and others described in our introduction relating to the S1, S2 FCS is in the context of various CoV spikes have led to speculation that the SARS-CoV-2 FCS could have been similarly inserted to characterize its function in different cell types. The virologist David Baltimore commented that these features make a powerful challenge to the idea of a natural SARS-CoV-2. The David Baltimore? Like the actual real guy? That's like a super popular. Wow. Still alive. Yeah, that seems a little weird. Yeah, that's not good. That's not. You shouldn't. He's just speculating there. Yeah, a month later, he said that he should have softened the phrase "smoking guns" because I don't believe it proves the origin of the fur either way. I believe that the question of whether the sequence was been naturally or molecular is very hard to determine, but I wouldn't rule out the origin. Yeah, it's hard to say that. Later clarifying that you can't distinguish between two origins that just looking at the sequence. Okay, so let's see what they say here about it. There has been intense discussion exactly how an FCS could experimentally. How, there's been intense discussion about how exactly an FCS would be experimentally inserted into SARS-CoV-2. For instance, some scientists have pointed out that the SARS-CoV-2 FCS insertion is out of frame. I mean, this is pretty nuts. I mean, that is crazy that it is out of frame. I mean, that's it's hard to explain how crazy that is, but basically the codons that are aligned in order to create things in a cell, you can have them match up in the triplicate order and the codon order that you normally would have, but this is a out of order means that everything, like imagine a sentence, but take out the first letter and move like the words are the same, but then the letters of the words are shifted over to the left. It would make gook, right? For instance, other scientists have pointed out that the PRRA insertion utilizes a CGG doublet that might suggest codon optimization. The CGG codon only appears with a 5% frequency in SARS-CoV-2 like viruses. Here, hence, some scientists have speculated the CGG doublet is improbable in natural SARS-CoVs. However, the rarity of a codon cannot be used to confidently determine the provenance or rule out the provenance of a given doublet. For instance, a similar PRRAR motif is in the spike of a feline cove has been observed to be encoded with a CGG-CGA for double arginine. In this case, a single mutation could change the CGG to CGA, CGG to CGG. Our position is that without access to the full set of viral sequences available to scientists prior to the emergence of SARS-CoV-2, it's difficult to know what range of novel cleavage sites might have been experimentally characterized and how these sites may have introduced that of SARS-CoV. As more bat coronaviruses are sampled, it is possible that other coronaviruses will be discovered with the S1, S2 FCS insertion. FCSs have evolved naturally in other non-cerbevoviruses families of coronaviruses, beta coronaviruses. Therefore, an S1, S2 FCS emerging in a cerbevovirus is consistent with natural evolution. Even so, the knowledge of that scientists had a workflow for identifying novel coronaviruses sites in diverse SARS-CoV coronaviruses and experimentally characterizing these cleavage sites in SARS-CoVs, likely in a manner that makes the resulting recombinant SARS-CoV practically indistinguishable from a rare SARS-CoV with natural emergent FCS, makes it challenging to rule out an artificial origin of SARS-CoV-2. Yeah, that's so weird. I mean, I think that they're just saying like that there's such a small sampling of the natural origins that they can't really make 100% accuracy statement. But like this, I mean, this definitely is different than anything seen in other papers, right? Which is kind of weird. and they kind of just have a, quoting David Baltimore there, just kind of like randomly. I don't know if I would trust this, just for, I don't know. It's just very weird. I wonder what the back of yeah. Let's see what I wonder what the other papers are saying about this, the more recent ones. I mean, we have so much information now, comparatively, and I could have. Reply to Gary. Jeffrey D. Sachs. Oh. This must have been it. Shan. This seems. So this is a letter to the editor talking about this. So this is Neil Harrison and Jeffrey D. Sachs. Where are these? Where's this lab? I'm confused. I'm curious, like, how other scientists. FCSs are common in coronaviruses and present in representatives of 4 out of the 5 beta coronaviruses subgenuses. We stated that the furin cleavage site in SARS-CoV-2 is unique among sarbevoviruses, although FCSs are common in beta coronaviruses. So this is the same alignment here. The FCS's PRAR and the furin cleavage site between R and S is indicated by the red arrowhead. Yeah, this right here. The highly variable nature of the S1-S2 junction is easily ascertained by inspecting a precise alignment of cerebral spikes. Gary's limited alignment of 7 spike sequences is far from precise. For example, lacks the arginine. A comprehensive alignment highlights the unusual FCS of SARS-CoV. Placing the insertion out of frame would be an unusual, needlessly complex feat of genetic engineering. Conclusion that the insertion is out of frame thus unknowing the template for the insertion. In any case, an out of frame insertion, for example, to engineer a restriction site is neither unusual nor complex. Yeah, I mean, it's possible, but why would you do that if you're trying to do any type of natural evolution, directed evolution experiments? The immutable proximal ancestor of SARS-CoV-2 first evolved in an intermediate host. This is complete conjecture. No intermediate host has been identified. I mean, now I think that you could say that. This is 6 months ago that this came out. 2 related lineages, lineage A and lineage B, first infected humans via the wildlife trade at the Wuhan Institute. What? This has been, this is completely wrong. So, 2 related lineages, lineage A and lineage B, first infected humans via the wild. No animal harboring lineage A lineage is identified by the Wuhan. Like, this is completely wrong. I'll show you the paper after that does that. 4 extra amino acids, not 8, were added. They were the insertion of 12 nucleotides into the spike. I don't agree with these people. Who is Neil Harrison and Jeffrey D. Sachs? Interesting. Yeah, I mean, I don't like, I'm just really skeptical here. Man, he's the director of the Center for Sustainable Development. Ah, man, dude. Like, these people are way too high up in order to be making these weird. Oh, man. These guys always like that are doing this stuff always have these weird paths. I'll be honest with you. I don't know, dude. Like, okay, so like this stuff right here, that wasn't the best alignment, you know? It's kind of like, I mean, there's a little bit better ways to kind of like display this information. So it's kind of weird to just copy and paste it. And let's just do it. Let's go to Werribee. 2 related lineages, lineage A and lineage B, first infected humans via the wildlife trade at the Wuhan market. So this is not correct. November 2022. I don't understand why he would say something like that. This is true. There were 2 lineages. No infected animal. How could you make such a definitive statement? I don't understand. This is still in preprint. It's under review. It's not going to be. It's been more than a year now, and it's a preprint. I doubt this gets to publication. And where is their genetic lineage here? They're saying that there wasn't things. So how did they test for? They did PCR and CT analysis. Okay. But that's not going for lineage. Reverse transcriptase positive number. Like qPCR data isn't what you would want to do. They're just using reverse transcriptase PCR. Show me the lab. The lab of what? Welcome to chat. Here's the stalls and the distribution of the positive, but they're like qPCR is going to be for the detection of like, yeah, I mean, I just, it doesn't really sit well with me to be using this paper. That's a preprint that in over a year, by the way. And even this one is showing 2 distinct genotypes. This paper even shows that there are distinct lineages. Genotype S and genotype L. Even the one that they linked. No infected harbored lineage A or lineage B was identified. Right here. This is the paper that they're citing. Lineage genotype L, genotype S. I don't understand. And then they're doing environmental PCR samples here. Very odd. It's honestly crazy that it's been that long in review, I'll be honest. Like, that's kind of weird. Especially if they're here. The viruses from the market share. In the summary, the study provided convincing evidence of the prevalence of SARS. This reprint, I don't know, man. The viruses from the market shared nucleotide identity. That's not really a good way of like doing it. You would do other ways. And even in their own phylogenetic maps, they did have two lineages. They're just not showing, say, in the abstract. And there was RNA found in this in new papers that have come out recently. So I think that this is being held in preprint because it just doesn't match with current data, and you're not. That's just doesn't match up. Doesn't make any sense. Cited by 44. Oh, this is so frustrating. How can a preprint get so much? It's just one of the really frustrating parts of being a scientist during this, where you have like data that just doesn't match up on these bioarchives and the preprints, and then people are like cite them as that, like, this is huge and this is a breakthrough, and you're like, it's not even peer reviewed, man. Like, what do you mean? People that are experts in their field don't agree with this. Doesn't sit right. This is disappointing. Okay. All right. I don't feel like that was really good analyses of the Furin cleavage site. I mean, they did make a good point in that paper where in Nagy et al. 2021, they did make a good point about the need for sampling, sequence sampling, and they did say that there needs to be stuff like, shown, and that does corroborate with the other papers that were saying that we need more in-depth sequencing, because the the SARS-CoV-2 virus had to have been, the precursors had to have infected a lot of people. So we do need more sequencing of that stuff. So that did align kind of like what other people are kind of saying, but yeah. So this is okay, so here's the Furin cleavage site. This is kind of like a basic paper. It's basically knocking out and showing that a Furin cleavage site is necessary in order to get transmission and infect the ACE2 sites, the ACE2 parts of the cells. So I don't think that's the growth rate. We can just, so yeah, they used a lentiviral SARS-CoV-2 polybasic insertion at the S1, S2 cleavage site that can be cleaved by Furin. Using engineered spike variants in live virus competition assays by measuring growth kinetics. Found the selective advantage in lung and primary human airway epithelial cells. Depends on the expression of the cell surface protease, which enables endosome-independent virus entry. Okay. SARS-CoV-2 virus lacking the S1, S2 furin cleavage site was shed to lower titers from infected ferrets and was not transmitted to co-housed sentient animals, unlike wild-type virus. So basically they put infected a bunch of stuff with SARS-CoV-2 infected rats that didn't have this part, and they didn't show any type of infection, unlike the wild type virus. Okay. I think that's pretty, that part was interesting in that, like, it had to and needs to be there. We know that in order for SARS-CoV-2 to be infectious, we have to have that furin cleavage site. But we kind of knew that there were that selective pressure that we were talking about before. Okay, back into actual published stuff. Okay, so this is August 2021. 4 novel SARS-CoV-2 related viruses were identified in the rhinophlid bats. Horseshoe bats again. Okay, so we're showing lots of bats, lots of horseshoe bats. And we're trying to figure out basically what recombined in order to make SARS-CoV-2. High diversity of bat viruses were present in a very small geographic area. That's really good for recombination, right? Because you have a lot of different parts of DNA kind of coming together and recombining. Okay. I think that there's a really cool graphical. So this is like molecular interactions between these two, the receptor binding domain. I thought that there was like a map. Oh, here we go. So these are all that type of bat that we know. Oh, geez, man. Oh, man. Is this an overlay of both of them? Is that what's showing here? Oh, man, that's a lot of them. 23 different types of coronaviruses. Oh, my God. So they are doing that genetic sequence and environmental sequencing that they were talking about. We collected, to reveal the diversity, ecology, and evolution of bat coronaviruses, we collected bat samples in the Yunnan province, China during 2019 to 2020. Damn, that is... Imagine the bravery. That is crazy. Overall, 40 of the 100 sequencing libraries contain coronaviruses, including 7 libraries with contigs that mapped with SARS-CoV-2. I mean, how do you explain that type of like richest diversity in everything in other than a natural zoonotic and spread? I don't understand why people are so entrapped and enveloped into this idea of a biotechnology origin. For 24 novel coronavirus genomes from different bat species, including 4 SARS-CoV-2-like coronaviruses, additional PCR-based tests reveal the presence of these 4 viruses in 9 individual samples collected in Huan province between May and July. Dude, they got right onto planes and were like, we need to go to China. We need to. That's right after we were the sequencing about the where it came from too. That is so cool. Together with the SARS-CoV-2 related virus collected from the Thailand in June 2020, these results clearly demonstrate the virus closely related to SARS-CoV-2 continue to circulate in bat populations and in some regions might occur at a relatively high frequency. A particular note was one of the bat coronavirus identified here exhibited 94.5% sequence identity to SARS-CoV-2, revealed that the genome as a whole and in some individual gene regions was the closest related to SARS-CoV-2 identified to date. However, much lower sequence identity in the spike protein, which is important, undoubtedly the product of a past recombination event, made it the second closest, next to rat G13. Hence, aside from the spike, RPYN06 possessed a genetic backbone that is arguably the closest to a SARS-CoV-2 identified to date. Although several SARS-CoV-2-like viruses have been identified from different wildlife species that display high sequence similarity to SARS-CoV-2 in genetic regions, none are highly similar, greater than 95%, to SARS-CoV-2 in the spike gene in terms of both the sequence identity and the amino acid residues of critical receptor binding sites. I mean, I don't think that that if we know that the mutations are going to be specific to the organisms that are accrued in them over time, I don't think that your hypothesis would be that you would have a furin cleavage site that or a spike protein that is so similar, because you need to attach to ACE2, human ACE2. Why would you have a human ACE2 in a bat coronavirus? Indeed, the spike protein sequences of 3 of the novel coronaviruses described here formed an independent lineage separate from the known Sarbo coronaviruses by a relatively long branch. In this context, it is interesting that the recently identified bat coronavirus from Thailand carried a 3 amino acid insertion at the S1 and S2 cleavage site. Yeah, we were just talking about that. Although this motif is different to that seen in the SARS-CoV-2 PRRA and this PAA, this once again reveals the frequent occurrence of indel events in the spike proteins of naturally assumed sampled beta coronaviruses. Come on, dude. Come on. Oh my gosh. Strikingly, these strains have only possessed 1 deletion in the RBD or be able to bind to HAC2, albeit very weakly. Accordingly, it is possible that there might be another lineage of naturally occurring circulating coronaviruses with spike gene sequences that confer a greater potential to infect humans. That would be gnarly. I guess it's just that they're not, maybe they're they can infect humans, but they're just currently infecting other parts of other mammals, or the ACE2 of those. Collectively, these results highlight the high and underestimated genetic diversity of the Sarbo virus spike proteins, which likely underpins their adaptive flexibility. Rhinolophid bats, those are the horseshoe bats that we were talking about before, are important hosts for coronaviruses. They're adapted to dense forest environments, and the majority of species rarely fly significant distances outside of forests, as their wing morphology and call frequency is better suited to cluttered habitat conditions. Small body size in rhinoflids mean that the GPS tracking is not possible for the majority of the species, although their adaptation to forest habitats make long-distance migration unlikely. While our monitoring efforts suggest that some larger bat species from other genera may migrate, seasonal abundance changes in species such as blah, 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 and all this showing peaks in April likely reflect seasonal roost changes from the region. Okay, so they're kind of talking about the migration patterns. While long-distance migration is unlikely to cause the spread of coves across the region, the possibility of novel communities of bats of highly similar species forming during key periods such as the breeding or following the loss and degradation of roosts could drive host shifts between rhinoflids. Previous studies have revealed frequent host switching of coronaviruses among bats. Okay, so that's what we needed before. Interesting. Indeed, we identified nearly 100% identical coronaviruses from multiple bat species, indicative of the frequent cross-species virus transmission. As noted before, this is in part likely reflects their roosting behavior and propensity to share the same or close habitats. In this context, it is noteworthy that 3 of the newly identified SARS-CoV-related gene coronaviruses grouped with the pangolin-derived coronaviruses from Guangxi is the whole genome phylogeny. In the whole genome. Although the associated branch lengths are relatively long, such that the host may be involved, there are topological differences between gene trees. This is suggestive of virus transmission between pangolins and bats. Wow, how would they interact? How would pangolins and bats interact? Recently, a new SARS-CoV-2 related coronavirus was identified from a pangolin from Hunan. Whether pangolin-derived coronaviruses established a distinct clearly warrants our ecological modeling revealed a high richness of rhinoflids across much of the Southeast Asia and southern China, with up to 23 species projected to coexist from the 49 species in analysis. The largest expanses of high bat diversity habitats stretch from South Vietnam into southern China. Indeed, it is striking that all the bat viruses described here, as well as the RMYN01, which is the one that we were talking about, is very similar to SARS-CoV-2, were identified in a small area in Yunnan province. This highlights the remarkable phylogenetic and genomic diversity of bat coronaviruses in a tiny geographic area to which humans may be routinely exposed. In addition to rhinoflids, this broad geographic region in Asia is rich in many of the bat families and other wildlife species that have been shown to be susceptible to SARS-CoV-2 in vitro. It is therefore essential to further surveillance. Okay. Interesting. I liked so embarrassing to these people. I don't understand why they would. Okay, so I liked out of tonight. It's a lot of papers. I think I'm gonna end stream here, but we like the host specific. This art. What is this journal? We didn't see this one. Yeah, I mean they're just talking about the mutations and the deletions that are It's in a really abstract journal. Archives of virology? Okay. Oh, no problem, bro. Identification of novel bat coronaviruses shed evolutionary light. Okay, yeah, this is the one. I like this one. Yeah, dude, bats are really crazy. That's an insane. I mean, 20, 30 different types of coronaviruses all in an area where they're super close to one another and they're exchanging respiratory. That's crazy. I didn't know. I thought pangolin or bats were kind of like where the genetics were mapping on, but now it's like specifically bats and these horseshoe bats, which is really interesting. Novel SARS-CoV-2 related coronaviruses in bats in Cambodia. So another one of these. I want to like learn more about these horseshoe bats. Okay. Structural modeling. Okay, so maybe they're talking about here the. Oh, this is a short paper, dude. Short. Ooh, so this is the recombination that we were talking about. Similarity. Dude, look at rat G13, just like right up there. So I don't. Okay. Wow. Okay. I think we can do one more paper. Knowledge of the origin and reservoir of the coronavirus responsible for the ongoing COVID-19 pandemic is fragmentary. This is Delaune 2021 novel SARS-CoV-2 related coronavirus in bats from Cambodia. To date, the closest relatives to SARS-CoV-2 has been detected in the rhinoflask bats sampled in the Yunnan province. That's what we just talked about. This is a good follow-up paper to talk about. Here we describe the identified SARS-CoV-2 relatives in bats in two horseshoe bats sampled in Cambodia in 2010. That's crazy. Metagenomic sequence. So this is trying to see basically where that sequence has been and what's been happening to it in that same area. Metagenomic sequencing identifies nearly identical viruses sharing 92.6 nucleotide identity with SARS-CoV-2. Wow. So this has to do with like that rat G13 where it's like 50 years ago this had to do with it. So this was in 2010. So how would so the person leaked it from the lab in 2010 in Cambodia. This is how stupid this idea is. It's just so frustrating. All this genetic evidence. Most genomic regions are closely related to SARS-CoV-2 with the exception of a region of the spike which is not compatible with the ACE2 mediated entry, the human ACE2. The discovery of these viruses in bat species not found in China indicates that the SARS-CoV-2 related viruses have a much wider geographic distribution than previously reported and suggests that Southeast Asia represents a key area to consider for future surveillance of coronaviruses. Dang. So they found 2 related SARS-CoV-2 bats in Cambodia in 2010, and wow. So this has nothing to do. Where did the other one do? The identification of novel background. It's just like, so this was this is in Cambodia down there. They did sequencing. Dude, imagine the conditions that you'd have to research in order to. So this is the Zhou 2021 paper, identification of novel bat coronaviruses. This is where they made these sampling. So look at that. Oh my God. They just hit it right on the money. Right here. Look at that richness, diversity. 23 different coronaviruses there. And right there, they nailed it. They basically, both of these papers like showing that area is as insane genetic diversity of these bats. And the ones that sliding window analyses of changing patterns of sequence similarity between the SARS-CoV-2 in China and Cambodia. They've C21, C45 were emerged for this analysis. Source data were provided as a source data. The phylogenetic tree of different genomic regions from left to right are regions spanning this, the spike and N-terminal domain, spike receptor binding. Okay, let's see the full-size image. So these are these CoV-C45s. And they're like right along with everyone. This is looking at different parts of the genome that binding domain that we were talking about earlier. Wow, this is a really interesting way of like showing these phylogenetics. This is incredible. They're looking at different parts of the spike and the orfs and all that kind of stuff, the different parts of the genome, and seeing which part actually aligns with which ones. You can tell the genetic diversity is like there. There's SARS-CoV-2. SARS-CoV-2. So again, rat G just really closely related to that pangolin right there. And then this is the CoV 45 and 21 here. Extensive evidence exists of numerous recombination events in the evolutionary history of these Sarbevo coronaviruses. Oh, that Sarbevo viruses is what SARS-CoV-2 are. I didn't know that. I thought it was just a coronavirus. Wow. SARS-related coronavirus, or SARS-CoV, is a species of virus consisting of many known strains. Okay, so the SARS-CoV-2 species is a member of the beta coronavirus of the subgenus Sarbevo. Oh, okay. So the subgenus is Sarbevovirus, and the genus is beta coronavirus. Okay, so it's just like the way that these kind of things are related to one another. Genus beta coronaviruses, subgenus Sarbevo coronavirus. Okay, that makes help. That helps. Consistent with this, we found that both RSH STT 182 and 200 are also mosaic viruses. Yeah, dude. There's tons of structural modeling of this. Okay. So a lot of the sequence diversity here. Here's that rat G13, SARS-CoV-1, SARS-CoV-2, and this is that GG next to one another that people were freaking out about. And there's tons of that diversity. I don't understand why people say such stupid things. Genuinely. Like, be a scientist, man. Like, what are you doing saying weird stuff like that? You're David Baltimore, man. How embarrassing. Nobel laureate again, dude. God. I think it's just like they think that they were right and they went against the in order to get their Nobel, they had to kind of go against the grain. So they're gonna like say something that they could just make up anything and kind of like they're too smart to do research anymore. Like, just doesn't make any sense, dude. Like, why could you make something so confidently? Okay, the data presented here further indicate that SARS-CoV-2 related viruses have such a wide geographic distribution than previously understood and likely circulate via multiple rhinolophus species. Current understanding of the geographic distribution of SARS-CoV-2 and SARS-CoV-2 lineages possibly reflects a lack of sampling in the Southeast Asia. Okay, exactly. Or at least across that region. Finally, pangolins, as well as members of carnivora, especially in the are readily susceptible to SARS-CoV-2 infection, might represent intermediary hosts for transmission to humans and should not be ignored for future surveillance in the region. Viruses of the SARS-CoV-2 sublineage, with one exhibiting strong sequence similarities to the SARS-CoV-2 in the ribosome binding domain. Right? That's what it means, right? Receptor. Oh my god, ribosome. God, I got fucking microbiology in my brain. While it's not possible to know where these animals became infected, it is important to note that the natural geographic range of the pangolin species involved also corresponds with Southeast Asia and not China. Southeast Asia, which coasts a high diversity of wildlife where exists extensive trade and human contact with wild hosts of SARS-like coronavirus, may represent an area to consider in the ongoing search for origins, and certainly in broader coronavirus surveillance efforts. The region is undergoing dramatic land use changes, and that's going to push stuff into human habitat. Like, way to go, guys. Way to go. Okay. So things are like super common in Cambodia now. 2010, 13 years ago, before anyone was looking into this area, we know that a very, very similar sequence exists that we can track. Okay. Do 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 do. Okay. A novel potential recombinant rodent coronavirus of the polybasic cleavage site in the spike protein. Man, it really goes to show like this pandemic just made people lose their minds. And it's so sad because all these people, I don't know why, like scientists like are hitching their with no evidence, just throwing out at like wild things like that. It just doesn't make any sense to me. It really doesn't. it's so flippant that they're making these comments that where it's like these things have big impacts, you know? And now it's just embarrassing. It's just embarrassing for them. They're never going to be able to live that down. I don't understand this just yet. I think that they're trying to look at the different types of recombination that's existed in rodent coronaviruses and seeing if that could be a repository. RARR in the spike protein, which is dominant. Interspecies transmission of coronaviruses and stuff. Interesting. So they're looking at different intermediaries sometimes. That'd be cool. Well, you know, not cool, but man, it's really, really frustrating. Okay. Virum characterization of game animals in China reveals a spectrum of emerging pathogens. Nice. $1,900 game animals from 5 mammalian orders were surveyed for viruses. 102 mammalian infecting viruses were discovered, 21 posing a potential risk to humans. Exciting. Civets. High number of potentially high-risk viruses. Exciting. Nice. And these might be wildlife reservoirs. I forget where. Man. I wonder. Well, this obviously has that wet market connection, right? I'm curious. I wanted to like kind of like go through the background because there's you know, 2 huge papers that basically we're not going to go over tonight because I really want to go over those in super depth. So I wanted to kind of like look at everything else tonight. Wow. These have remarkable symmetry for combination and analysis. It's crazy. Dang, look at all that sequencing. God, that's crazy. That's so cool, man. That is so cool. Why would there be these viruses that look so similar to SARS-CoV-2? Like think about that for a second. Like why would there be so many viruses in this area of the world, exactly where the zoonotic spread happened, that have so similar sequences to the one that we have? Like like come on, man. Come on. It honestly it's not hard. Our study therefore highlights the fact that coronaviruses are subject to relatively frequent host jumps among mammalian species. Oh my gosh. So I wonder if they have happened. Okay. Oh my gosh. Another back coronaviruses later cause infections to humans. Wow. So we got to go through all of these. Exciting. All right. So we have. A lot of papers still to go over. Dang. Ooh. What was this one? A novel potentially recombinant erode. Oh, that's right. That we already went through that one. Dude, it's on like this is just so crazy. The amount of science that was going on. So cool. Oh, this is a review. Dang, we could have just read that. That would have been easy. What is this? I am afraid that we must adjust our lives to COVID-19 for many more years from now. The best case scenario would be probably natural immunity, meaning that most people on Earth will finally be immunized with natural infection. This would indeed be the final point of the pandemic, but new strains can arise and unknown virulence able to reinfect humans. Don't forget the viruses HIV, Ebola virus infect humans for decades, and it still are too virulent. So yeah, I mean, those are still pandemics, technically. We are dealing with a huge viral spread. How is this a mirror? How did this get? Where is it? The best you can do is invest in vaccination strategies. Wow, that's. This is so funny. This is the weirdest paper. It's so flippant. Here, I would like to borrow the knowledge you gained from other pandemics of the previous century. Like, this is nuts. Very interesting. Who is this guy? Very like flippant. I don't know how to explain that language other than like very casual. Cool. You nailed it, dude. Dude, that's so funny. I'm afraid SARS-CoV-2 is here for a while. He's the senior editor of that journal. No wonder he just like wrote that in an afternoon and got it like published. Like, like what the hell, man? What the hell is that? That's not a review. That was just him like shooting the shit, man. Dude, you should have to say whether or not you're an editor to the journal. I'm sorry. I'm afraid we must adjust our lives. Dude, people are dying, bro, and you're like afraid that you're gonna have to like adjust your life. Like, what is this? I think for constructive proofreading of this paper, no funding department, no ethics. He's the editor of the journal, man. He's the editor of the journal. It's huge conflicts over. How can you say that? That is nutso. Nutso. Okay. I think we're gonna take it. Dude, there's recombination that exists all the time. I don't understand why. It's hilarious to me that people don't think that it's possible. I just don't. Dude, this is a cool study. Oh, this is from fucking this is from track. Oh no, it's like next strain. I know that. Oh no. COVID covariance. That's it. Dude, you just like putting covariance as a certain figure, bro? He's just doing covariance? So this is like nextstrain.org is a cool way to track all the different phylogenetics, all the different changes. See, this is like, dude, this is not a figure, bro. This is oh, it's performing on NextJ. Okay. All right. We'll allow it. That's just funny, man. I was looking at this. I've been looking at this stuff for so long. To actually see it in a paper is pretty funny. You can actually see the mutations by codon where it is. It's so cool. Look at all the spike, all the spike mutations. Look at this new one. Wow, that really took over. Global all time. Wow. Whoa, look at that genome. That is so cool. Wow. That's so cool how they just go up right up to there. And then they get outcompeted again. The same thing. Man, that's cool stuff. Okay, I think that's worth reading. Okay, I think these are the ones that I'm going to read in the future. 1. The origins and evolutions of SARS-CoV-2. I think this is worth reading. Okay, we're going to do that. Picard, obviously, we're going to do this. These are like, okay, I mean, you don't, spoilers. All right, man. All right, later, dude. Have a good one. Okay, so this is what we're going to be covering next week. So these are really cool papers, man. They're like getting down to, like, we know like pretty much exactly where even which stalls this all came from. The map of the Wuhan market and stuff. We know so much data now from this. So this idea that like we didn't know 2 lineages and all that stuff, it's like, what? Like even like there's 2 lineages right here. Like even that one paper, the paper that they were citing, there was 2 lineages. You just like what are these people talking about? They're embarrassing themselves as scientists, like, but they're providing no evidence, and this is the evidence, right? That's the thing. Like, in order to make a disruptive claim and say that something's happening, okay, show it. Like, show your data. Make a hypothesis and test the hypothesis. But a lot of times they don't. They don't want to. And that's the problem, is that it's lazy and it's contrarian just to be like contrarian. And it's embarrassing. It's just absolutely embarrassing. So these are the papers that we're going to read. So let's hit it up with some virome characterization of this, because this would be a good paper to read before Picard, because this includes Picard and Orabi. Yeah, that's cool. This includes sequencing of these unknown birds and raccoon dogs. So they actually went in there and sequenced that stuff after the pandemic. So it's really cool. Okay. Exactly. Wildlife trade is likely the source of SARS-CoV-2. That's what it is. Sorry. That's what it is. That's what the evidence is. It's true. This is March. This just came out. March. This is kind of like a little. Sorry, guys. Little spoilers. This is Warby before that, but that's a good. This is a good idea to put it out. Damn. Okay, and then that coronaviruses. Okay, I think those 4 are going to be awesome material next week. Okay, so we went through a lot of really good papers. We found some really cool papers, and now we have a good idea of what to do next week. I think that's a great way to leave it. It's very late, and that was fun. Thank you all for watching. These research streams are a little ad hoc. I think the idea is to kind of like show the idea is to kind of show the research process, right? Where new ideas come from, and like researching something. I think it's cool to kind of show this stuff unedited and to show the actual process of actually doing research. And this is research for me because I've already searched through a lot of this stuff in order to find these papers. And I'm familiar with them, but now I'm searching through them again. Or I'm researching. And that's what it is, like reacquainting yourself with it, contextualizing it with new data, with new papers, with new sources and new materials and methods. And that's what science is, just tireless pursuit of trying to figure out what's happening. And as we saw tonight, like there's some stuff that people are proposing that just doesn't make any sense. And I'm happy that we saw that, actually. I'm really happy that there were some things that came up and we could kind of just like talk through it and be like, this doesn't add up with all the other things that we were talking about. And there's a reason why they're preprints, right? They're in these bottomless holes of publications and journals where they're never going to see the light of day, except for people that are trying to artificially hold them up as something that they're not. And you look at data that we looked at, you look at all the ways that people have investigated where this thing has come from, and it's obvious. It's obvious where the data is, where the reproducibility is, where the story lies, because that's where all the data is. That's where the reproducibility is. There's where all the sequencing data for the bat coronaviruses for the last 50 years has come from, for example. So, and I love the diversity of methods that we looked at today, from molecular biology, RNA evolution kind of stuff, looking at mutation rates and the chemical evolution of, the genome itself, to more just standard sequencing techniques, right? And just broad sequencing stuff. And those two methods have to come together, right? They have to coalesce into a similar story. Otherwise, someone's uncovering something that's never been before found. And that's not what we're finding here. We're finding that they are telling the same story, the same different research groups across the world that have different ideas and different motivations and different financial backings and different scientific backgrounds are all coalescing around the same idea of where this thing is coming from. And it's not a lab. So I still see no scientific evidence that this came from a lab. So That's what it is. That's what it is. That's what it is. All right. Thank you so much if you watched any of this, or if you have any people in your lives that are questioning this. I don't know why they would watch. How many hours is this? 2.5 hours. But maybe you tell them, hey, this is some of the evidence to go. I think I want to like succinctly put this into one presentation and make a video of it, but I want to show the research process. I think that's really important. That's one of the things that I'm trying to show with this channel, is that to destigmatize or kind of make more transparent the actual research process in science and the background research. The background research. We're not actually doing research here. We're reading things and establishing like a hypothesis, and then looking how other people are testing it. But that's what happens. That's what happens. A lot of reading, a lot of kind of understanding, trying to understand what's going on. And I don't understand. I don't understand everything that's every single sentence or something, but we look up Wikipedias and we try to contextualize it and we try to talk it out. And that's why it's fun. Learning. Always learning. Always something to learn. And man, that's why I love science. Science is so cool. Okay. Thank you so much. I appreciate you. This is the peer review with Greg. Thanks for tuning in.